Dr. Krishna R Murthy. The next paper, potential biomarkers in the urine of babies with ROP.
Good afternoon, everybody. I don't have any financial interest. The retinopathy of prematurity is a potentially blinding disease which often affects premature low birth weight infants. It's the 5th leading cause of bilateral childhood blindness worldwide. And the principal risk factors attributed to this disease include prematurity, low birth weight, and oxygen supplementation. The disease is characterized by an abnormal proliferation of the developing retinal blood vessels at the junction of the vascularized and avascular retina. Screening for retinopathy of prematurity, the gold standard for this is binocular indirect ophthalmoscopy. However, with the advent of digital retinal photography, telescreening models are fast gaining importance. In this study, we explored the possibility of finding urinary biomarkers for ROP using high-resolution mass spectrometry based quantitative proteomics. These biomarkers, once validated, can be developed into point-of-care kits, which will take away the screening from the ophthalmologist or the camera to an NICU nurse who can look for screening directly in the NICU once these biomarkers are validated. Briefly, let me describe what proteomics is about and what entails in a mass spectrometry-based experiment. Proteomics is the systematic study of the protein complement of a cell. It encompasses an effort to catalog and define functions for all the proteins which are there in a cell. This slide gives you an overview of a mass spectrometry-based proteomics experiment workflow. Let me first describe the components of a mass spectrometer and the principles involved in it, and then I'll take you through the workflow of the experiment. A mass spectrometer has an inlet, an ion source, a mass analyzer, an ion detector, and a data analysis system. The analyte, which needs to be examined, is first introduced through the inlet, and when it reaches the ion source, the molecules of the analyte are converted into gaseous ions. These ions, when they enter into the mass analyzer, are separated based on their molecular weight and detected by the ion detector, and the data is then analyzed by the software. The proteins are first isolated and extracted from the tissue of interest, and it is subjected to enzymatic digestion to yield a mixture of peptides. The enzyme commonly used is trypsin. The peptide mixture is then further fractionated and subjected to a mass spectrometry-based analysis, and the data is further analyzed. In our experiment, urine samples from premature babies who developed retinopathy of prematurity and those who did not develop retinopathy of prematurity were compared. The urine samples were concentrated by passing them through a 3 kilodalton filter. The protein amount in each of them were estimated and normalized. An in-solution digestion with trypsin was done, and the samples were then labeled with TMT. TMT stands for tandem mass tags. These are isobaric tags. Basically, they have unique molecular weights. So once tagged, the mass spec is able to pick up these based on their masses, and you can go back and see what happened actually to the sample to which it was tagged. The samples were then pooled and subjected to basic reverse phase liquid chromatography-based fractionation and examined on the mass spectrometer. This is a pictorial representation of the experiment. Control samples and ROP urine samples were first trypsin digested, and then they were tagged with isobaric tags. The samples were pooled, subjected to basic RPLC fractionation, and examined on the mass spec. A total of 1,336 proteins were identified in this experiment. 20 molecules were found to be differentially expressed across 12 pairs of comparisons. By 12 pair, I mean 4 ROP samples and 3 controls. Each one was compared against each of the controls. So ROP1 to control 1, ROP1 to control 2, ROP1 to control 3. So you had 12 comparisons, which by itself is a self-validating process. There was more than a 2-fold increase in the abundance of 8 of these proteins across all 12 comparisons. So these are potential biomarkers for us. This is a list of the 8 proteins which were significantly elevated in the ROP urine samples. And this is an expression trend of the overabundant proteins in the ROP urines. You can see here that the level of the protein, and this is derived out of the mass spec. You can see here that the level of the proteins are much higher in the ROP samples in comparison to the controls across all 8 proteins. To conclude, this study provides a number of proteins which are found to be overabundant in the urine of ROP patients. We are now validating these overabundant molecules in a larger cohort of ROP samples using the ELISA method. We intend to use these biomarkers for the development of inexpensive and easy to use point of care devices for the detection of ROP. I would like to acknowledge here the work of my colleagues who have helped me with this experimental process. Thank you. So these were with ROP which went on to needed treatment. So these were vision threatening ROP, the ones which needed treatment for the pilot study. Yeah.
And the other persons who, where ROP regressed, did you separate them out? The people where ROP did undergo regression?
No, they were not seen as a separate group at all.
They were as good as no ROP?
Yes. All of these were photographed, no. So the controls didn't have any form of ROP at any point in time. So they were not the ones which missed out. Each one of these were photographed through RetCam. The ones who went on to develop vision-threatening ROP were the ones who were there. The ones who were controls did not have any form of ROP at any point in time.
Krishna, wonderful study. I would like to know whether these babies were gestational age matched, weight matched, and at what point of time the urine samples were collected. Was that also matched?
No, they were not matched in the pilot study. What we are doing now is going to be matched for all those. These were just taking diseased to normals and looked at that.
So I think because these are these things can cause a lot of variation in the amount, the proteins that you are measuring.
Including comorbidities, actually.
Yeah.
Those also needs to be looked at.
And that's what I was wondering, like if there were other features in the preterm where these proteins were also raised, and will you consider all those minor factors which lead to kind of progression of ROP?
Correct. Very true. So the what we are doing now is actually looking at sequential samples along with all the other data that we have, 2 weeks, 4 weeks, 6 weeks, 8 weeks, and also looking at photographs next to it and looking at the protein levels of the 8 proteins. So these are basically leads for us on which we can work now on a larger cohort with this statistically significant in a field trial, and also look at it in a sequential manner, whether it precedes, whether it comes after the disease or before the disease. Everything can be mapped now in the ongoing study which is happening.
No, like if that child had septicemia or other things, so these proteins will also be expressed in those. It's like you will take comorbidities into consideration.
Comorbidities will be taken into consideration, but as long as it significantly gets elevated with your disease, it still is a biomarker. Doesn't matter from where it is coming, because it's not going to be causative, it's going to be only a passenger or a indicator.
So that would be a confounding variable. Like if they had comorbidities which were causing this, proteins to be, secreted in the urine, like what Dr. Dinesh Garg said. So that might be a confounding variable,
It will be, but if you take it away from the pathogenesis, if you say that with disease is it going to be there, then it doesn't matter from where it is coming, as long as it's always associated with your disease statistically, then it still becomes a biomarker.